Do it. 50 or 30. Boy, the battery life on my computer is now 5 years old, and it still works great, except like its battery life is not what it once was. Does anyone have that with like a computer in your life? Yeah. It's getting older than the battery. She does not hold up like she used to. Battery does not hold up like it used to, but that's okay. All right, so we're going to take a little break to talk about eukaryotic evolution. In other words, we know fungi are eukaryotes. How did we get eukaryotes? Early life was prokaryotic. And prokaryotic means that the cells had no nuclei or organelles. Well, this, of course, raises the question, how did, bless you, the nucleus and the organelles evolve? And we don't know for sure. We don't know for sure. But our best evidence, guys, my interest in talking over top of people is very limited. Shh, thank you. But our best evidence is that nuclei and other organelles evolved by a process called endosymbiosis. Endosymbiosis. Hello, welcome. Okay, let's break that word down. When you see the prefix endo, what does that mean? Creatures endogenous to a region, and endoskeleton, yeah. Very good, inside. Excellent. Endo means inside. And what's a symbiote? What's symbiosis? Symbiosis from grade 9, yep.
It's kind of like when an organism feeds off of another organism.
Yeah, but not feeds off of because there's two kinds of ways in which that can work. And one we call parasitic, right? So a parasitic relationship is where one feeds off the other. And symbiosis is where they feed off each other, not necessarily for nutrients, though it can be, but in mutual benefit. So for example, you've seen those fish that latch onto sharks, and they clean living things off the skin of sharks, so they keep them clean, and they get rides around, right? So everybody wins. Or the birds that eat the food from between alligators' teeth. How'd you like that to be your living? I'm just going to land on this alligator's mouth and eat the food out of its teeth, and the alligators are cool with it. They let them do it because it keeps their teeth clean, right? That'd be way better than flossing, wouldn't it? Just open on up and let those bad boys get at it. You should try that. Anyone got birds at home? Just see what happens. Anyway. Yes.
So then would a covalent compound be considered symbiosis?
No. No, I refuse to accept that connection to a different class. You're not in chemistry class right now, you're in biology class, dude. I mean, maybe, I don't know. Anyway. Okay, so endosymbiosis means inside a mutually beneficial relationship. What the hell does that mean? Right? Well, let's take a look at it. This is the theory. Guys, in all seriousness, I need it to not be that hand down. I need it to not be that every 2 seconds I'm asking you to be quiet. Let's just focus in. So, the basic idea is this. Imagine that we have two cells. A larger cell. And this would be a prokaryotic cell. All right? We got a cell. That cell's got some DNA floating around, right? And then let's say it does what cells do, which is it's hungry. And cells normally eat things by a process called endocytosis. Endocytosis. Endo, inside of, cytosis, cell. They consume nutrients by bringing them inside, endo, the cell, cytosis. Does that make sense? So, we have a large prokaryotic cell, And then down here, I'm going to draw a small prokaryotic cell. But to the large prokaryotic cell, what it's seeing down here is dinner. Does that make sense? Hi, how are you? So don't draw this, all right, ready? But what normally happens is you have big cell wants to eat this thing. And what happens is, if the thing it's eating is cellular, is it bumps right up against it, and then it can actually engulf it, like this. Don't draw this. And it can end up with the thing inside, and then it can attack that, break it apart with all the various machines it has in the cell, and consume it. Does that make sense? And steal its nutrients. So this, don't draw it, is a process called endocytosis, bringing something into the cell to destroy it. Does that make sense? And the best theory we have is that the way we get organelles is that the large organism consumed the small organism, all right? Just like it normally would. So this is absorbing the smaller cell via endocytosis. But then something interesting happens, which is, instead of dying, the small cell survives. And in the billions of times that a larger cell consumed a smaller cell for nutrients, our best theory is that a few times this happened, where you end up with a cell inside of a cell, basically. Does that make sense? And over time, sorry, I was going to draw this splitting off. That would even persist after cell reproduction. Basically, that cell inside the cell could reproduce, as could the larger cell, and so the offspring of the original cell would also contain the offspring of the other cell. And depending on what the arrangement was, this could lead to a few different ways. Can you name some of your organelles in your cells, in the human cell? Yeah. Mitochondria. All right. What does the mitochondrion do? Yeah. Gives you energy. Specifically how? Does anyone know? We take nutrients that we've eaten and we actually put them into the mitochondria, where what happens to them? Does anyone know? Yeah, they basically get burned. Like literally burned. They react with oxygen. And they produce carbon dioxide as a waste, water as a waste, and they produce energy the cell can use. Just like a fire in your backyard. Literally, a mitochondrion is an engine. It takes nutrients, but the thing is, before the mitochondrion, bacteria did this same thing, and they still do today. Bacteria and our ancient ancestors, prokaryotic cells, do that same thing, but they do it in the cytoplasm. Does that make sense? It's one of the many machines in the cytoplasm. But that means that the food just ends up spread throughout the cytoplasm. These machines end up spread throughout the cytoplasm, but in your cells, there's a place to go. There's the mitochondria. And the theory is that this is how we got the mitochondria. It just so happens that one day in the past, a big cell ate a small cell, and that small cell was really good at consuming nutrients and making energy. Does that make sense? Much better than the big cell. And it survived in the big cell, which means that over time, most of the nutrient processing happened not in the big cell, but in the small cell, which means that over even more time, the genes for nutrient processing in the big cell stopped being used because they weren't needed, because all the nutrient processing could happen in the small cell. And we actually have good evidence for that. Your mitochondrion has its own DNA, and it replicates itself. It is a self-sufficient cell inside of a cell, which is kind of cool. Interestingly, the DNA in your mitochondria, the DNA in your cell, half of it comes from mom, half of it comes from dad. The DNA in your mitochondria, all of it comes from mom. Because your egg cell had mitochondria, the sperm cell doesn't put all of its organelles into the zygote, into the forming egg, just its DNA. And so all of your organelles are descendant of mom, including your mitochondria. So when we want to check, one of the things we can check, mitochondrial DNA gives you a sense of just the maternal parent line. It goes just down your mother's. So your mother, your grandmother, and to your mother's mom, to your mom's mom's mom, all that, sticking with the maternal side all the way up. We can get a track of your maternal ancestry by following your mitochondrial DNA, whereas your main DNA covers all of your ancestors. Your mitochondrial DNA, only the mothers. And only mom's mom, only that mom's mom. It sticks down just that line. So it doesn't branch out. Your mitochondrial DNA points to a straight chain all the way along the mothers of your family tree, which is kind of cool, right? I think that's kind of cool.
And does the DNA stay like 100% like the same as the one before?
Good question. That's actually one thing we can tell when we look back at things that are a few thousand years old, is we can sort of rank, part of how we can figure out their age is that DNA naturally does change over time. And so how similar the mitochondrial DNA can give us a sense of where in the family line they land. That was a great question. Yeah, naturally degrades and changes and will mutate over time. So you would expect that you and your mom should have pretty much near identical mitochondrial DNA. It should be identical or maybe half of 1% different. But if you went to your great grandmother, it might be 0.25% different. And if you went 1,000 generations before that, it might be 2 or 3% different. So it's changing very slowly, but we call that genetic drift. Good question. So, what evidence do we have for this? The first piece of evidence is that all organelles are membrane bound, just like a cell. So different parts of the cell are made of different things, but all of the organelles are surrounded by membranes. And that implies that the mechanism of how they would have come to be by cells getting absorbed into the cell is consistent with that. That doesn't mean that's not proof for this, it's evidence, right? It suggests this. 2, some organelles still have to this day their own DNA. An example of that is the mitochondrion.
So this only happens when the mitochondria or with all human beings?
Sorry?
Does this only happen because I know you said like they're not organized?
No, we'll talk about this. The theory is this is how they all came to be.
So is only from mitochondria?
No. Mitochondria is one example. Okay. So the next example we're going to look at actually is the nucleus. Now contains the main DNA of the cell. And the basic idea there would be that at some point, the reason DNA is kept inside the nucleus is that it is profoundly better protected. Does that make sense? Much better protected. So once the evolution was happening where some of the DNA was kept in the main cell and some inside this new cell inside of a cell that we call the nucleus, the DNA in the main cell would get damaged at a much faster rate and degraded at a much faster rate, but the DNA in the main cell would still work, and they could still be making all the stuff the cell needs. And over millions and billions of years, the DNA in the main cell actually got wiped out. It wasn't necessary. Does that make sense? So the theory would say that, according to this theory, over time, it became not necessary to have any DNA whatsoever in the cell itself, and all of the DNA was in the nucleus. The upshot of this theory. Does anyone know what upshot means? Because it doesn't mean what it never means what it sounds. I always think upshot means like it should sound like the advantage of, like the good thing about this, but really it just means like what this means, like the natural repercussion of this. The outcome of this theory is that organelles were cells with strong specialized functions that were absorbed by endosymbiosis. I'm going to say were brought into the cell by endosymbiosis. So the function of the nucleus is that it protects the DNA, right? It is much better at protecting the DNA than just being loose in the cytoplasm. We already said what the mitochondrion was good at. What was its function? Yeah. Mitochondrion is energy from nutrients, right? And again, eventually, most of the mechanisms to do that in the main cell died out. So if you take grade 12 biology, you'll see that some nutrient extraction still happens in the main cell, in the cytoplasm. Most of it takes place in the mitochondria. What's another specialized organelle that has a clear function that you know about? Thinking plant cells here. Cell wall is not considered an organelle, though it's an important structure. But that would have evolved differently, not by this. Sorry? They do. Yeah, ribosomes are necessary for reading DNA. Chloroplasts, the ribosome is also not considered an organelle, though it's related to the organelles. It's a structure. It's a cell structure. It's not membrane bound, and it's not sort of its own thing with its own plasma inside. It is just a structure. The chloroplast is able to photosynthesize. So you can imagine, just think about this. Imagine if you could do this today. Imagine if your cells could absorb a plant cell and steal its chloroplasts, such that over the course of a couple of weeks by eating plants, all of a sudden you start turning green and you could photosynthesize. Wouldn't that be incredible? Like if you're like, oh, I'm pretty hungry, and you just went out and just stood in the sun and you're like, now I'm full. Blood sugar spiking. Be crazy, right? Like, wouldn't that be amazing? Never have to eat again? You're stuck in the desert, be like, oh my God, I'm so full. I just can't stop making energy here, right? But could you imagine? Now, obviously, it doesn't work that way, but you can see how absorbing a cell that could photosynthesize into your cell, real advantage. Because what don't you have to do anymore? Eat nutrients, right? Instead of eating that cell once by enslaving that cell and making it do its job in perpetuity, not only inside you, but also its offspring inside of your offspring, you're able to never have to eat again. A reminder that nature is a brutal and mean place, and plants are basically chloroplast slave owners, which is horrifying. Like, seriously, if you think about it. But that's what this theory presupposes, that basically it captured a cell that was good at photosynthesizing and said, oh, you're in here now, do your thing. And they do. Yeah. The endoplasmic reticulum is an organelle. It's an organelle that attaches itself to the nucleus, and its specific function is actually processing mRNA. You don't need to know this till grade 12, but if you're curious, the DNA never leaves the nucleus, but it actually gets used out in the cytoplasm. So how do you do that? And the answer is you make a photocopy, right? Just like we won't let your student record leave the office. But if you need a copy of your transcript, they just take your transcript and they make a copy of it, right? In the nucleus is the blueprint for your cell, and every second of your life, copies of genes are being made and put out into the cytoplasm to be used. Does that make sense? So the main copy is kept safe, and the endoplasmic reticulum takes that mRNA and it processes it before it's used in the cytoplasm. That's its job. Yeah. Yeah. So the idea is that at one point, there's a cell floating around, big cell, it's hungry, and there's this little cell floating around. That little cell happened to be really, really good at making energy, better than most cells, really effective, really has lots of machines in it to be really good at taking nutrients in and making energy. When it got eaten by the cell, 99.999% of times it would just be eaten and destroyed and its nutrients used. But one time, it ended up inside the cell and it didn't die, it stayed there. And all of a sudden we have a symbiotic relationship where this cell inside of the main cell can absorb nutrients, and instead of doing what it normally would have done, which is process it itself, it would get absorbed by the small cell inside, which would do a way better job of absorbing those nutrients. And so that, being an advantage, hung on. And then because that small cell can reproduce inside the big cell, all of a sudden this big cell has a bunch of small little cells inside of it, which we call organelles, which are very good at processing energy. And when it splits, some of them end up in this daughter cell, some of them end up in that daughter cell, and now all of its offspring have their own little cells inside of a cell, which we call organelles, which are good at doing this job for them. The nucleus is another example of the same thing. The endoplasmic reticulum is another example of the same thing, the Golgi apparatus. It's all these little cells that used to live on their own that now live inside of another cell that are kept alive because they're good at the thing they do, and they've evolved along with the big cell over time. That's the theory. We don't have direct evidence for this. We don't have direct proof. We haven't photographed it happening, but that's what our science suggests to us is what's taking place. The dominant theory is the theory of endosymbiosis. Yeah. We've done this artificially, yes. If what you're asking is like, have we tried to make this happen? Because yeah, we have, and it works. You can definitely get a cell to live inside of another cell. Yeah, no, there's all kinds of experimental techniques going on with exactly that kind of stuff. I mean, we evolved and adapt before that, but this is certainly a big step in that, right? And when we do our whole unit on evolution, we'll talk about sort of how evolution specifically works, but this is definitely an example of evolution, but it wouldn't have been the first example of evolution, no. Evolution at its most basic started from our earliest ancestors, where random change leads to diversity. And that's all evolution really is. Random changes are happening, and over time, every cell isn't the same anymore because these grandchildren of the original cell have developed differently than these grandchildren, and so where you used to have just cells that were all identical, now we have different types. That's evolution. And if you let that happen for billions of years, the differences get bigger and bigger and bigger until you have trees and people and carrots and bacteria and fungi, which all are cousins of each other, but very different from each other. That's right. Yeah. Because your cells don't have one mitochondria necessarily, they often have many.
But how does the big cell not get overwhelmed?
Because again, I'm sure sometimes they did, in which case it would have just died, right? So important to remember, we're not seeing all the times this failed, right? So yeah, if it absorbed it and it just kept reproducing and eventually caused the big cell to burst like a virus, well, that didn't get passed on because the big cell's dead. So maybe that happens 99% of the time, or maybe even 99.999% of the time this happens, but we don't have evidence of those because unsuccessful adaptations don't get passed on, right? If something kills you before you reproduce, then you can't pass it on. So what we're seeing is the time that didn't happen and the time it worked. And we don't have a way of knowing, did it work most of the time and only fail sometimes? More likely in biology, what we find is most times things fail. most adaptations just kill people. Most mutations just kill people. Most things are negative. Because you imagine, if you randomly change stuff, you are way more likely to break something than to make a new thing, right? So most random mutations harm an organism. But every once in a while, they don't. Every once in a while, you get a trait that is novel and beneficial. Did that answer your question? It was a really good question, so I hope that made some kind of sense. I'm a professional communicator, but I often feel like a professional confuser. But that was okay? All right, good. Yeah.
You majored in bio, right?
I majored in bio. Well, talking about what I majored in is, I majored in health sciences, then a brief foray through philosophy, then briefly history, then chemistry, and then biochemistry, and then through a paperwork error upon graduation where I filled out the form wrong, my degree says that I got a degree in biology, despite the fact that I finished a degree in biochemistry. So do with that what you will. My university years were circuitous. I didn't take a straight line. I didn't know what I wanted to do.
How long did it take for you to
I did it in 4 years. I did it in my 4 years, but I was here, there, and everywhere. And I had to take some night classes and summer classes to get caught up when I changed degrees. But yeah, no, I did it in 4. But I wouldn't recommend my path. I say that it made me well equipped to be a teacher because I studied a little bit of everything, right? So that would be a bad path if what you were planning to do was something specific. But with what I do, where I have to teach a little bit of everything, I actually think it helped me because I have a pretty well-rounded education, though not as deep as I would like. People who studied biochemistry and that was all they ever studied really got a deeper education than I have. So you want to talk chemistry, Alastruck knows a lot more than I do because he was much more focused than I was. My education was different. It was pretty broad. I think it makes me well-suited to be a high school teacher, though it does sometimes lead to embarrassment where I find myself going ooh, that's something I should know, but I don't. But it's like anything else in life. You trade one thing for another. Yeah.
Is this the last one?
Oh. I don't know if you know this. It's still September.
I meant the unit.
No, we got a few. We're going to go through the kingdoms. All right. So that's the idea. So then from there, one branch of the eukaryotic family tree is fungi. Hey, there we are. No, of course not. Okay, so fungi are complex eukaryotic organisms. I'm going to do a quick, don't worry, I will come back to this. Don't freak out. All right, do we promise not to freak out? Everyone promises? Just relax. Yeah, okay, settle down. So I just want to show you, we're going to draw this ourselves, but we want to look at a basic fungal cell. And a basic fungal cell is fairly similar here, but you'll note that we have mostly similar structures to what we have. The large difference is twofold. One, you will note that they have a cell wall. And then you will notice as well that they reproduce differently. They don't reproduce like we do, like bacteria do by binary fission, nor by pure mitosis like we do, but they engage in this process of budding, where they section off a septum, a small section of the cell, and the next cell starts to grow there. So instead of growing and then dividing into two equally sized cells, fungi tend to grow by sectioning off a small portion, and that grows into a separate cell, typically, though not always, still staying connected. Does that make sense? So we're going to note that here. We'll just draw a cell. Okay. How much of that do I want to include? I don't want to include much. Okay, we could have a vacuole, which is basically a big storage unit. We could have the nucleus, which we know what the nucleus contains. The nucleus contains DNA. On the inner lining of this, we have the cell membrane, which is surrounded by a cell wall. Very good. We have the mitochondrion and other organelles. And as they are growing and reproducing, we can have the bud. And the bud will have its own nucleus with DNA, its own mitochondrion, its own vacuole, it's all those things. So it's mostly the same organelles as humans. And plants. Some even have chloroplasts. Some fungi reproduce. We'll mention that here. Though not most. Say they have a cell wall. And they reproduce by budding. So the new cell starts as a small offshoot of the parent cell. And the structure that forms between them is called the septum. A septum is just tissue that separates two things. So, like you talked about the septum of your nose, right? Some people get the septum pierced. The septa in your heart that separate the chambers. So this is called the septum. And the septum forms and slowly, so basically these things would grow, move up there, and then the septum would form, and the bud would eventually split off to its own cell. Bless you? Question mark. I didn't know. Do you say what? I don't know what you say to a hip route. Okay, separate. We can do this. Separate. That E looks. Somebody look that up for me.
A-R-A-T-E.
A-R-A. Yeah, see, I should have trusted myself. Look it up. You have my blessing to pull out a device to check my spelling. Now I'm embarrassed.
Do fungals, if they have chloroplasts, do they photosynthesize?
Some. Yeah, some do. Not most, but some.
Yeah, aren't some considered plants sometimes?
Not considered plants, so they're now single-celled. Fungi. All right. So these include yeasts. So single-celled fungi including yeasts, can exist. An example of this would be, let's see if I can get this right, Saccharomyces cerevisiae. Okay, the odds that I'm going to correctly spell Saccharomyces cerevisiae are very low, so I'm going to quickly Google that, if that's okay. Does anyone object? I say I missed the H. Does anyone know what Saccharomyces is? Saccharomyces is one example of a yeast known as brewer's yeast. But this is the yeast you use if you are making wine. It's the yeast you use if you are making beer. It's the yeast you use if you are making bread. It is absolutely everywhere in the human condition. We use it for all sorts of things. This is, bless you. You all right? Don't trip. This is what it looks like under an electron microscope, and you can see the individual yeast cells, and you can see them budding. Note that they don't only have one bud necessarily, right? See how this one is budding in several places? But Saccharomyces cerevisiae is really noteworthy because we use it so much. And do you know why we use it?
Why?
Because it has this special characteristic where if you give it a bunch of sugar, it will metabolize that sugar quickly and start to produce gas, CO2. Really quickly. It'll eat sugar really fast and make CO2. And it just so happens that if you want to make your bread light and fluffy, if you just take flour and mix it with water and bake it, you basically get a rock, a brick of like imagine if you left pizza dough, like when pizza dough won't rise, or if you've ever had like a Seder meal and had like unleavened bread, you just get this kind of thick like consistency. But if you put yeast in there, they will start to eat the sugar of the bread, even more so if you add artificial sugar, and they will start to produce gas. And inside of a dough, that will make little pockets of air. And what does that do to your bread? Makes it rise, and it makes it all nice and light and fluffy. Then you put it in the oven, and now you have delicious, light, fluffy bread. What has happened to the yeast, by the way? Oh yeah, you done killed it real good. So basically, you take yeast and you say, hey, here's a bunch of food, mix it up in a dough, and it's like, whoa, this is great. We got a bunch of food. Let's get eating. And they get eating, and they produce gas. And then you say, hey, into the oven with you, and then they're dead, and your bread is nice and light and fluffy. And then we discovered, or maybe we discovered this one first, I'm not sure which one, we discovered that if you give it a bunch of sugar and then you deprive it of oxygen, if you starve it of oxygen, if you suffocate it, in a desperate bid to keep surviving, it will undergo a process called alcoholic fermentation, where it will still metabolize that sugar, still produce CO2, but in a desperate bid to survive, to get rid of its waste, it produces a waste product called ethanol, which is a toxin, but we know it more commonly under the name alcohol. So when you have wine, what you're having is you got a bunch of grapes, you shove some yeast in there, then you put it in a container where the yeast couldn't get any oxygen, and you let it desperately try to survive. It produced a bunch of alcohol while trying to survive. Eventually, the alcohol killed it because alcohol is a poison, and you're like drinking that and being like, well, this is delicious. Thank you, Saccharomyces cerevisiae. So we basically use and abuse this small microorganism because we enjoy what it can do for us. Because just like nature, we are vicious beings, humans. Vicious we are. Yes?
So when you use active yeast, is the foaming it budding or is it...
CO2.
Yeah, I was gonna say
Yeah, it's
that.
just producing CO2. Yep.
So the yeast we use for alcohol the same as the yeast we use for like
Yeah.
store?
Yeah, so to be clear, like if we get to it in 4U biology, I usually do a lab where we make alcohol, right? Like where we take yeast and we get some sugar and we deprive it of oxygen. And then, if time permits, we'll go into the chemistry lab and we'll separate the alcohol from the everything else, right? Like, yeah, it's, there are different strains, just like there's different breeds of dog. They're all the same species. By the way, how did I do writing this binomial nomenclature? What does it need? Two things are wrong. Both should be underlined or italicized. I'm going to go with underlined for obvious reasons. And the first word, the genus, should be uppercase, and the second one, that looks ambiguous, but should be lowercase. So that's better, right? This is bread yeast. Used for baking and brewing, whether you're brewing beer or wine or liquors or whatever.
Is that what breweries?
Yeah, you'll sometimes see it called brewer's yeast as well. So those are single-celled fungi. Then we also have multicellular fungi. Now, you could take an entire course or several courses on this. I did. I took 2 years of fungal mycology in my university degree, which I found very interesting. I do not have time to teach you everything about it. We're not going to learn our Picanidia from our whatever, but we're going to learn the most basic, which is this. Multicellular fungi, fungal cells, grow in a vast network. Not a vest. They do not grow in vests. I mean, they might grow in vests, so make sure you wash your clothes. But they grow in a vast network called a mycelium. Of tube-like cells called hyphae. Okay, 2 words here. Mycelium, which is the network, and the cells themselves, which are called hyphae. I don't have to do anything. I feel like I did close the bracket. Yeah, call the mycelium close bracket. No, that's okay. Listen, keep me honest. But like a broken clock, I get to be right twice every day. And this happens to be one of them, team, and you got to be here to witness it. There you go. Okay, let's focus. So, to show you what that looks like, though you've seen what it looked like, because you saw it over there, but to show you what it looks like here, a hypha is the singular, hyphae is plural. So this is what, if you look at your cells there under the microscope, this is what it looks like at a big scale, fuzzy. See how it looks fuzzy? The reason it looks fuzzy is those hair-like filaments are actually strands like this. Can you see that? Pretty cool. And each of these is a singular hypha. All right? And then, yes, you can get this structure called a conidia on a conidiophore, which is reproductive. But the basic structure, is these long tube lips. And the interesting thing is that they can actually connect to one another. They stay connected. They have what's called a cytoplasmic continuum, or cytoplasmic connectivity, through what we call septa. So, again, if you look at a structure here, that's a better image. These are individual cells, individual hypha that are connected. I get all excited talking about this. It's interesting stuff. So, at a large scale, or at a human scale, fungal mycelia, which is the plural, appear fuzzy or hairy. At the cellular level, we see this. So that would be zooming in on one of the little hair-like structures of a fungus. So it's got all these little hair-like projections there, right? And if you zoom in on just one of them, this is what you'd see. Does that make sense? That's the world's worst diagram. But does that kind of make sense? So the whole structure here is called the mycelium. And each of these is a single hypha. The plural is hyphae. And the singular here is mycelium. That L should not be there. Or sorry, the plural is mycelia. You'll note that the septa between cells are porous, which means that the cytoplasm of one is connected to the cytoplasm of the other. Does that make sense? Now you'd have organelles in there, so you would have, a nucleus with its DNA in each one, presumably. Most cells have a nucleus. Every once in a while you get one that doesn't. You would have mitochondria. You'd have vacuoles. You'd have all the things that cells need inside each one. Let's do a vacuole. Whoa, big old vacuole. Ooh, big old vacuole. Okay. And you'd have your endoplasmic reticulum and your Golgi apparatus and all that jazz. Does that make sense? Yeah.
Is there a reason why hyphae was lowercase but hypha?
No, no. It doesn't mean anything. I just was drawing attention to it. Would you prefer that I write lowercase like the others? Then it would match. I'd kind of prefer that. I like when things match. No. I like when things match. Okay. Nope, no time. Don't worry, it'll still be here. Multicellular fungi reproduce by producing spores. And these are at branches off the hyphae called conidiophores. Or on the fruiting body of the fungi, which are typically known as mushrooms, etc. When you see a mushroom, the mushroom is a fungus, but it's only a part of a fungus. Specifically, it's the fruit, the fruiting body, which again is different than the fruit of a plant because the fruit of a plant contains seeds and the fruit of a fungus contains spores. But that's what that fungus is. That's what the mushroom is. And if you tap it, which means when you see a mushroom on the ground, you're not seeing most of the organism. Where is most of the organism found? Under the ground, in a vast network of fungal mycelia, of hyphae. Hyphal cells work through the ground. So where there are mushrooms, if you dig down, and if you have a little sample kit and you grab a microscope, you will find hair-like structures underneath that mushroom. That is the fungal hypha. That is the organism itself. And it can be enormous. It can spread for meters in every direction. Yes? Where? This?
No, at branches off. I just don't know what that's supposed to
Yeah, at, like as in the spores can be found at branches, on branches. On would be better. On would be a better word. on branches off the hyphae called conidiophores, or on the fruiting body of the fungus called mushrooms. You know? Yeah. Yeah, in a moment. So I want to show you. So does anyone know the species of the common white mushroom that we use in cooking? Is the noble Agaricus bisporus. Isn't that a good looking? Look at that, the Agaricus. So the thing of it is, the mushroom is on the surface, but underneath the surface is all of the mycelia. So button mushroom on the surface. So the Agaricus bisporus, I'm going to shroomer.com. That can't get me in trouble. What a nice diagram, though. Yeah, so if you look at that, you can see, look at all the fungal tissue here, and the mushroom is just the exposed portion of it that is used for reproduction. Not all hyphae have to have a large fruiting body, but one that is underground does, because otherwise how will its spores spread? If it releases its spores under dirt, they're not going to get very far, right? If it grows up onto the surface, especially with this, because it's evolved a brilliant thing. It's evolved something that animals like to eat. And if an animal eats the fungus, or eats the mushroom, and goes out, and not to get graphic, but then it will defecate, and the result is that those spores will have been moved somewhere else, but those spores will also be moved somewhere else and deposited on the ground, surrounded by fertilizer. Pretty good deal, right? So all you got to do is make it, which is exactly what plants do, right? Plants have evolved to put their seeds inside a structure that you find delicious, so that you will eat them, and go deposit that seed in a big pile of manure somewhere. Now we, as we always do, like to screw nature over by putting it in a wastewater treatment plant and irradiating it and killing it. But animals at least do what plants want. Does that make sense? The sugar and fruit and stuff isn't to feed the seed. There's nutrients inside the seed for that. It's to feed the animal that it's hoping will carry that seed off somewhere else and let it spread out. Does that make sense? It's an evolved technique. Here we go on shroomer.com. Yeah. So next time you have mushrooms at home, if they get to the point where the cap underneath is starting to degrade, tap them on your countertop, and you'll see little like dots, basically. Each of those is a fungal spore. Yeah. Yes, oh yeah. Mushrooms like cold, dry conditions with lots of fertilizer. So not to be crass about it, but if you mix some dirt with some shit and some spores and just leave it in the dark, it will take care of itself and you will get massive mushrooms, which is exactly what they do at mushroom farms. Do you know what Ontario's third largest cash crop is? Mushrooms. Crazy. We're a massive producer of mushrooms for the world. Yeah. Yes. How's that? Is that good? All right. Oh, yeah.
Yeah, she'll come
Sure.
down.
Yeah, you bet. Aiden, this is way better. Aiden, in the future, can you just be me when they page? Awesome. You have to sound like less impressive. You have to be like, put some ahs and ums in there. There you go. Now you sound like me. That's good. And those R's have got to kind of slur into L's, so they're kind of bit of that, like that kind of mushy mouth thing I got. See if you can do it. Let's hear it. Go. Be me. Go.
I can't.
Okay, work on that, all right? I want to hear it.
I'll take
Okay, if I get I have slash gun, I just need like to do 2 more things on the dichotomous key, and I'm gonna work on it next weekend.
What I'm hearing is it's done and you're turning it in, and when I check today, well after school is over, I'm going to find it totally done.
Maybe.
I think you're going to.
It's a 75.
25?
There we go. 20.
I think you'll be done. I have faith in you. And you can stick around if you need to.
Okay.
What's up? Which one? Do you know which ones? The which one? Yep. Yep. Great. I'll get both those for you for tomorrow. Okay. I'm having printer issues right now.
So, do you know what lessons? I think I'm missing a lesson for biology.
Do you have your notebook? Can we take a look?
Can I come after school and do some laser?
Yes. Yeah. 100%.
I don't think we have enough wood.
No, we'll get what we can done today, and we'll kill it tomorrow. Okay. And again, no penalty. Like, I appreciate that you're being ambitious. I will never punish somebody for doing something ambitious. I like ambition. You guys are good to go. Have a wonderful day. Yes, my friend. Yeah. Oh, really? Yeah, please. I love your copper band, by the way. That's really cool. May I? Yes. That is really cool. Oh, wow. So you were alive, because you would have been born what? You're 2010?
2009.
2009. So you were alive at the same time as your great-grandfather. Yeah. And great-grandma. Wow, that's really cool.
No, that's, I don't know what. Yeah.
Oh, okay. Well, that's really cool.
So, yeah.
Oh, very neat.
I do want to show you something.
Yeah, please. Okay, talk to me when. Be realistic. Pick a day and be realistic with me, and we'll give you an extension. So, but I want you to be realistic. So, what do you think? Thursday? Can we do Thursday? Okay. Thursday is your date. Okay.
Mr. Orlando, can you say the word relish for me?
Relish. Stain Chen Norway. Lived a good long life, eh? That's what. So this is your new discovery.
Kind of.
Very cool. Oh, that's so neat. And so this is Ancestry.com, eh? See, at some point in my life, I'm convinced I'm going to get really into Ancestry. That's really neat. Thank you for sharing. That's really cool. All right, let's take a look. Let's take a look-a-roozy. And obviously, like, don't stress about that assignment. We'll either not do it or we'll get to it later or whatever. We'll figure it out. How you doing in general?
Good.
Yeah? Nice. Was your summer good? Was it relaxing?
Not really.
No? Oh, no. Not good or not relaxing? Or not either? Oh, I'm sorry to hear that. Well, I hope you have a better school year than you had a summer. Now I got to figure out where I put 1.1 and 1.2. Let's just copy these. I'm having OneNote issues at the beginning of the semester.
That's so good. Wait, you're peer tutoring for this class? No, I'm in this class. I'm peer tutoring your class. Same grade. And Sunena is peer tutoring this class. And
Yes.
she's taking biology. Did she not? I think she did.
I don't know. Doesn't matter.
I thought she did last year. She took 3D bio.
I don't know. Anyway, I'm not worried about it. If I was in a plane that was crashing and Sunana got behind the wheel, I'd feel fine. And like, I didn't even know the planes don't have wheels until this very moment. But regardless, I'd trust her. Wouldn't you? Sunana's flying your plane. You're gonna land. She's gonna figure it out, right? Yeah. Right? She's a smart kid. She's a bright cookie. She's ambitious. All right. So you know where to look to get them filled in, right? Online. I've added you to this team, right? You're in there. Yeah, good. So we got 1.1. 1.2, 1.3, 1.4, so it's 1.5 that I'm missing, which is viruses and prions, which is handwritten? No, which is a handout. So you need this handout. Great, I'll get that tomorrow for you. Is that okay? You got 1 and 2 to work on, and I'm having printer issues. Is that okay? Sorry, I feel like a jerk.
Is there any assignments you want to say?
Not that I'm worried about. If you're feeling up to it, we can have you do this taxonomy one, but if not, we can just exempt you from it and move on. There'll be plenty of stuff to do in this course. You can do it? Okay, have you seen it online? I'll show it to you. It's fine. And we can make it due next week for you. That's fine.